Alright, this is part 2 of our gene expression lecture. And here, after we've reviewed all the basic structure of a gene, let's look at the process of transcription, how we turn that gene into RNA. And that's actually a little bit of a misstatement. We don't physically turn the DNA of the gene into RNA, we just read it and make an RNA copy. So let's start off by looking at all of the parts in transcription. We need first what are called transcription factors. And we actually have two types of transcription factors, what are called basal and specific. And be careful, don't confuse this with the Italian spice. That is the correct spelling there, basal with two A's. Basal means like background or basic. Okay. We'll get into what those are in just a sec. We need an RNA polymerase. And be careful, this is different from the enzyme we saw previously. Previously we saw DNA polymerase. DNA polymerase makes DNA. RNA polymerase makes RNA. We need our gene that we are going to transcribe. Specifically, we need only half of it. We just need the DNA template strand. Notice I'm not saying here parent strand. That's DNA replication only. And then finally, we need a thing called the spliceosome. I know that name seems like it came straight out of science fiction, but it's a real thing. So the process of transcription is going to be roughly the same in prokaryotes and eukaryotes, although some of the names will be a little bit different. Specifically, if you take microbiology, you'll learn how the transcription factors names are a little bit different in bacteria. Okay, so let's take a look at these transcription factors. Okay, in order to start transcription, we need these transcription factors. Let me draw you a picture of these transcription factors. Let me actually draw you a picture of a gene first. So here's, let me use a different color. Here's my regulatory regions of a gene. If you have no idea what I'm talking about, please refer back to part 1. Here's my coding sequence of a gene. Okay. So transcription factors will be produced by the cell, and they will start assembling themselves around these regulatory regions. So I'm just going to draw the transcription factors, little red blobs, attaching themselves around these regulatory regions. And if you remember, these regulatory regions, they came under a couple different names. A promoter was the most important one, and so that's this one right here. And then in eukaryotes, we can also sometimes have some others, and they often come under the names enhancers and silencers. Alright. Prokaryotes mostly depend on just promoters, mostly. So these transcription factors, they come in a lot of different flavors, right? There's a lot of different types of transcription factors, and you can get just a couple transcription factors assembling around these regulatory regions. You can get a couple dozen, right, or anywhere in between. So the amount and the specific type of transcription factors that you have determines how much a gene is read and how much that gene is expressed. In general, though not always, in general, the more transcription factors, the higher the expression level. What's really cool is that vitamin D actually acts, along with another protein, as a transcription factor. I think, do I have a picture of it? No, not quite. Okay. I'm going to come back to this in just a second, use this as an example. So if you recall, just from the previous slide, in eukaryotes we have two major groupings of transcription factors, what are called basal and specific. Often, under the name for basal transcription factors, you may see it in another textbook, They're sometimes also called generalized. These specific transcription factors are sometimes called activators and or repressors. I'm just going to keep it easy though and call them specific and basal. So let me take a little detour. Let me talk about these specific and basal transcription factors, okay? Let me give you some examples too. So here's a basal transcription factor right here. This is what it looks like. These transcription factors are proteins, so they look like proteins. They attach to DNA at the regulatory regions. So here we can see a DNA molecule that transcription factor is attaching to. The name of this particular transcription factor is called TBP. Stands for Tata box binding protein. We won't get into why it has this crazy name, Tata box binding protein. The important part here is that you need TBP present in all of your cells at all the times for transcription of any gene. TBP is a basal transcription factor. Remember, basal means baseline. So you need basal transcription factors in order to do any sort of transcription. Here, the next one is a specific transcription factor. You need specific transcription factors only for certain genes at certain times. So these specific transcription factors we really use to regulate our luxury genes. Remember those guys? Our luxury genes? The genes that we only need sometimes? So here's one example of a specific transcription factor, FOXP2. And FOXP2 stands for a long name. It stands for fork head box protein 2. Right? Not something you need to know. But notice he's also binding to DNA. He's binding to a regulatory region. And he's only produced in cells, we think, that are involved in language. So he's produced in certain cells of the lungs, certain cells of the voice box, and many cells in the brain that help to process language. In fact, animals that do not have FOXP2, whose protein is misshapen and can't bind to DNA, or missing, they don't speak. They don't have vocalizations. So again, not every cell, you don't need every cell in order to speak, so this is considered a luxury gene, or this is considered a specific transcription factor for luxury genes. So we need specific transcription factors for luxury genes. Here's another example of a specific transcription factor for a luxury gene. I love this one. This transcription factor is called Sonic Hedgehog, and yes, it was named after the video game character. This transcription factor is involved in expressing the proteins involved that help to pattern your body's symmetry. So the reason why you have 2 hands, 2 legs, why you're symmetrical, is in part, at least, because of this transcription factor being expressed while you are developing as an embryo. We don't use this transcription factor now, right? At least most of us, and most of our cells don't. So we've shut it off. Therefore, it's a specific transcription factor that's only present at certain times and in certain cases. So again, basal transcription factors we need all the time to express any gene. Specific transcription factors we need only in certain cases. So, now it probably makes a little more sense here. Depending on how many of these specific or basal transcription factors we have, that will determine how much we express this whole gene. And I know I'm leaving out a lot of information here. I'm just trying to keep it simple. Okay, so how these come together then? In order to express a gene, let's look at a luxury gene as an example. In order to express this luxury gene, we need both our basal and our specific. Remember, basal is for everybody, okay? Everybody likes basal. I'm trying to think of a way to turn that into a pun about the spice, but it's not working. Or the herb, I should say. Whatever. Okay. So right here, we're seeing 3 regulatory regions. This regulatory region is the promoter, the coding sequence is somewhere over here to the right. It's just left out of this image. Here's 2 additional regulatory regions. Those are enhancers. And some genes will have 2 enhancers, some genes will have 4, some genes will have none, some genes will have an enhancer and a silencer. Just depends. Okay? So here we see around the promoter, we have our basal transcription factors. Remember, general or basal, they are the same thing. I got this image from a different textbook, which is why the names are a little bit different. Around the activators, we have our around our enhancers, we have our specific transcription factors. Remember, activator, another name for activator is specific. So what happens is our basal transcription factors will use the help of another big protein. Here it's called the mediator complex. Don't worry about that guy though. Our basal transcription factors will interact with our specific transcription factors. So basal plus specific. And only then can you attract the big guy, RNA polymerase. So only once you get the proper combination of basal and specific transcription factors can you finally do transcription. Alright, oop, a little too far. There we go. So let me backtrack just a little bit, okay? Just a little bit. Go back to that vitamin D, just because it's such a cool example. Erase this stuff so I can make some room for myself. Whoop, too far. Okay. Man, I usually love my Surface, but sometimes it's a pain in the ass. Alright, so let's discuss how vitamin D works. You may know that we need vitamin D in order to absorb calcium, right? So we need vitamin D in order to absorb calcium from our diet. Here's why. Let's take a look at the calcium ion transporter gene. So again, zooming out really, really far here, conceptually, here's a cell. Alright. Here's the cell's nucleus. I'm making the nucleus really, really big so I can draw some stuff going on in there. And here's calcium floating along outside the cell. Let's say you just drank a glass of milk, so you got calcium floating along outside your cells. Remember, calcium is an ion, so it can't just spontaneously enter your cells, right? It can't cross the cell membrane. We have to use an ion channel to get that calcium across. Now, you don't get calcium all the time, right? So you don't constantly express these ion channels. They're only there sometimes. They're only there when when you have calcium in your bloodstream. Alright? So what I'm about to present is a very simplified version of what's going on, but it gets the point across. So we need to make these calcium ion transporters right there. Let me label these calcium ion transporters. Alright, so what vitamin D does then is when vitamin D is also floating along outside your cells, vitamin D will get inside your cells. Vitamin D, if you remember, is a steroid hormone. Let me redraw. So vitamin D, because it's a steroid hormone, it can get easily inside your cell membrane, and it can attach itself to a intracellular receptor inside the cell membrane. Okay, so here I'm drawing vitamin D attaching to that intracellular receptor. And that combination of vitamin D and its receptor will travel inside the nucleus and attach itself to DNA. This vitamin D and its receptor are just the right shape to be able to bind to DNA. And they will only bind to DNA of a certain sequence. Specifically, they're going to bind to an enhancer region So let me highlight the enhancer region. Oop, enhancer. Enhancer. Keep doing that. There we go. They're going to bind to the enhancer region of the calcium ion transporter gene. So let me draw this out over here now, okay? Let me actually do this. Give me a sec just to fill it in. What color? Okay, let's do red. So right here, I have my enhancer. I have my promoter. And then in purple, oops, I have my coding region for the calcium ion transporter. Alright, so far so good. Well, I'm going to start by assembling these basal transcription factors in and around my promoter. And I have many basal transcription factors. I actually have about a dozen. And these basal transcription factors are almost always going to be made by the cell. Because your cell is constantly doing gene expression, we need basal transcription factors for all of our genes, so these guys are always hanging around. Now, we need not only basal transcription factors to read this gene, we also need our specific transcription factor. And as it turns out, that combination of the vitamin D receptor and vitamin D is a specific transcription factor. So that specific transcription factor and the basal transcription factors together are enough to attract our buddy RNA polymerase. RNA polymerase will only start reading a gene once those transcription factors are present. So here's RNA polymerase. If you just have basal transcription factors, not enough. You just have specific transcription factors, not enough. The only time that specific transcription factor and the basal transcription factors are going to be present is when you have vitamin D. So, once everything is in place, RNA polymerase will read the coding region and produce an mRNA. That mRNA will go through a couple more steps, and we'll see what those steps are in a little bit. And we will produce the calcium ion transporter protein. That calcium ion transporter protein will be embedded in the cell membrane, and that's what allows calcium to get inside our cells. And there you go. This is a closer look, a nice computer animated version of how this works, if you want to go watch that. Okay, so we've got this set up. Now we know that we need both basal and specific transcription factors to attract our RNA polymerase and get transcription going. Alright, so that's step 1 and 2 that we just covered right here. Transcription factors are produced, they bind to the promoter and other regulatory regions, and then RNA polymerase will bind to the transcription factors. And here's the cool thing about RNA polymerase. By himself, he is able to untwist DNA, he's able to read DNA. We don't need helicase, we don't need topoisomerase, none of that other nonsense that we needed in DNA replication. Alright, so RNA polymerase, now he leaves behind the transcription factors and he will move down the gene. He'll read the DNA template strand and create mRNA. And remember, the only half he can read is the half that goes from 3 to 5. So let me show you an image here. Here's our RNA polymerase. We don't see any of our transcription factors in this image. They've been left out here for the, I guess, the sake of simplicity. Here's the promoter. We've left out any enhancers, again, for the sake of simplicity. Here's our coding region. It's also known as the protein coding region here in this image. RNA polymerase is going to unwind DNA, and that small little bubble right there That's called a, guess what? A transcription bubble. Shame on you guys who said replication bubble. An RNA polymerase will start reading the template strand. Remember that template strand goes from 3 to 5. And creating complementary mRNA off of that template strand. So he's reading the template strand every single nucleotide and laying down a complementary RNA nucleotide, starting at the 5' end and going 3'. And he's going to go all the way down the length of this gene until he hits the very end, until he hits transcription stop. Which means that where he started was transcription start, right? So at the end, he will fall off and release his completed RNA. And remember, it's a special type of RNA called mRNA. So that's what was all this gunk on the previous slide. mRNA production starts at the transcription start site and is manufactured 5 to 3. We hit a transcription stop signal, or transcription stop site, on that template strand, releasing both the DNA and the mRNA. Notice behind him, he's cleaning up his mess, he's rewinding the DNA. Here's a closer look at what actually happens inside RNA polymerase. Alright, so here's our template strand in dark blue. Let me get a dark blue pen. Template strand in dark blue. The non-template strand, the strand that we are ignoring, is in light blue. RNA polymerase is moving from left to right in this image. He will unwind 10 to 15 nucleotides at a time, expose them so he can match up complementary RNA nucleotides C to G, right, T to A, etc. And he'll rewind the DNA behind him. So he does the job of helicase and DNA polymerase all by himself, if we were talking about DNA replication. And here, this is actually a really cool image down here at the bottom. These are two genes side by side. Here's the beginning of one gene. Here's the end. Here's the beginning of the next gene. And here's the end. Alright, each of these little strings coming off, that's a piece of mRNA. So we have dozens of RNA polymerases all working on one gene all at once. It's not like the water, a water slide at a park, right? Where they put one passenger on and make sure that passenger gets all the way to the bottom before the next person gets on. Nope. One RNA polymerase will load on, he'll get going, then the other RNA polymerase gets on right behind him. If you've got those transcription factors there, right, you gotta make hay while the sun shines. Okay, so remember that type of RNA produced is mRNA. Okay, and the mRNA, it's the exact same structure as all the other types of RNA. It has ribose, it has AUCG as its nitrogenous bases. We just call it mRNA instead of tRNA or snRNA or siRNA just because it does a different thing. RNA polymerase will make a copy of the DNA template all the way through the entire coding sequence. And how thoroughly RNA polymerase attaches to its gene is based on the number and type of transcription factors that are present, right? We've been hammering this home. And specific transcription factors are often produced as a result of cell communication. We saw that with vitamin D, didn't we? So these, just to give you a couple other examples, these specific transcription factors, especially if they're steroid hormones, I'll come back to this in just a sec, especially if they're steroid hormones like estrogen and testosterone, they themselves actually help act as transcription factors. That's exactly how vitamin D worked. Vitamin D was a steroid hormone. It itself actually did kind of bind to DNA. Other transcription factors, specific transcription factors, may cause the expression of growth factors after a wound. So as an example, when you wound yourself, you release a molecule called PDGF, stands for platelet-derived growth factor. Platelet-derived growth factor triggers your cells to go through mitosis. Remember, growth factors trigger cells to re-enter the cell cycle. And PDGF is produced as a result of transcription and translation. PDGF is produced as a result of certain specific transcription factors triggering that transcription and translation. Other transcription factors may cause us to create glucose transport proteins after we've eaten a meal. Right? So we change our gene expression depending on what happens to our cells, and that's through controlling these specific transcription factors. And these are, by the way, this is all just for luxury genes. Housekeeping genes are a whole other thing entirely. Okay, so in prokaryotes, that's it. Transcription is done. In eukaryotes, it's a little more complicated. So I'm going to leave the complicated stuff, the mRNA processing, what happens in eukaryotes, till the next lecture. Thank you as always for sticking with me. I shall see you next time.